Hello everyone and welcome to another biology test review video. I believe that this is the review for test 5, but it might not be because we've done so many I've kind of lost track. As always, I do talk a little bit slow because there's only so many words that I can say in 1 minute, so please feel free to put this on 2 times speed and watch it much faster. Alright, today we're going to be talking about meiosis, genetic inheritance, and predicting genotypes and phenotypes, including using the Punnett square, dihybrid crosses, all the good stuff about genetics, right? There's a lot to say about this, so let's just strap in and go for it, right? So we're going to start right off with meiosis. So our first statement on, next to me I have the paper of statements that we need to know, and our first statement is the halving of chromosomes number allows a sexual life cycle with the fusion of gametes. So the purpose of meiosis is that it is necessary in sexually reproducing organisms because it creates the gametes, which are either sperm or egg cells, and those gametes each have a half set of DNA. I put it up here, but we can see that 1/2 of the DNA will come from the sperm and 1/2 of the DNA will come from the egg, and together those will make one full set of DNA, which makes a lot of sense. So first we need to compare sexual and asexual lifestyles. So I don't have this written down, but I will talk about it just verbally. Asexual reproduction is when an organism clones itself, meaning that there's only one parent involved. There is no reproductive process with meiosis. Meiosis is just not involved, and there's no exchange of genetic material. And the only way that genetic variance can happen is through mutation. So therefore the population will be less diverse. However, sexual reproduction involves two parents. DNA is inherited through gametes, so you get half of the genetic material from the mother and the other half from the father. Whereas in asexual reproduction, there's only one parent, so you get 100% of your DNA from that one parent. This sexual reproduction does require meiosis, either sperm and eggs or pollen and ova, like either of those. And there is a lot more genetic variance, and we will talk about them later. The next two things that I want to make sure that I am defining that I didn't have on this list previously is haploid and diploid. Okay, so haploids are going to be a single set of chromosomes. So this is going to be the gametes, are going to be your haploid cells, right? So again, that's the sperm and the egg. So the diploid cells are going to be everything that isn't a gamete, because every other cell is going to have a full set of DNA except for your gametes, right? So there are going to be two sets of chromosomes. And then I'm adding on here that with haploid, that's going to be 23 chromosomes. And with diploid, that means 2. Di means 2. So diploid is going to be 46. So it's two sets, one from the mother, one from the father. It's going to be 46. In addition, I have on here, so let's just go back to our statements. I'm all over the place. Explain why meiosis must occur as a part of a sexual lifestyle. Well, we've kind of already talked about this, but because asexual reproduction can't happen, then therefore meiosis must occur because sexual lifestyles require two gametes from different parents, each with a half set of DNA, right? Great, moving right along. Our next statement is one of diploid nucleus divides by meiosis to produce 4 haploid nuclei. So we will talk about this later on with meiosis, but you're going to start out with one cell, and that's eventually going to divide into 4 gametes. So that's going to be really a part of your haploid situation, right? So let's talk about the definitions that I have on the screen before we move forward. Some of these will come up in the statements later on, but let's just start here instead. Homologous chromosomes. So these are going to be two chromosomes that are the same. So they carry, in theory, the same genetic material, right? Like it's going to be the 21st chromosomes or the 22nd chromosomes. Like we have 23 chromosomes, if you didn't catch that already. And so the chromosome number 2 might pair up or whatever, right? Bivalent or bivalent, I don't know. When it's of homologous chromosomes, it's just going to be meaning that they're associated in pairs. So this will come into play when we start talking about meiosis I and meiosis II, but this just means that they are paired together. The chasmata, or chasmata, I don't know, is the point at which crossing over occurs. So this is going to be important because crossing over is going to play a big role looking down through the rest of the content. So let's just keep a big star next to that one, and we will remember that for down the road. A synapsis is the pairing of two chromosomes. Again, very similar to bivalent, except that's to be associated in pairs. This is to associate in pairs, I'm pretty sure. I could be wrong. This is a lot of vocabulary that we really only mentioned once or twice in class. And then the final main thing that I have here is non-disjunction. So when homologous chromosomes do not separate properly, and we will talk about that a little bit later. So let's continue looking at our statements. I'm gonna go through and mark down the ones we've done, so that means I don't miss any. So the next one is going to be drawing diagrams. I'm not going to be drawing diagrams because I think we all have brains. We all watched the mitosis video, and if you haven't, make sure you do, because that's going to be a little bit important to understand meiosis. Anyways, I digress. You should be able to draw diagrams. Moving right along, we're going to ignore that for now. I'll talk about meiosis I and meiosis II in just a bit. DNA is replicated before meiosis, so let's just do ourselves a favor and move on to the next thing. So, meiosis I, since apparently it just really wants me to talk about meiosis I and II right now. Prior to meiosis I, prior to prophase, this is going to be interphase. We know the deal. The DNA is going to duplicate. So this is going to provide us with 46. DNA is replicated before meiosis, so that all other chromosomes consist of 2 sister. So normally our DNA is like this. This is how our chromosomes look. When we duplicate our DNA, we get those pretty X's. This is going to be sister 1, or they're going to be sister chromatids, basically. So this is how our DNA looks. We get one set from our father, one set from our mother, right? Like we know the deal here. We're going to duplicate that. So we're going to have 2 sets of 40, or we're going to have 46. I don't know. Okay. Sorry. Anyways, the DNA will duplicate. Great. Prophase I, the DNA is going to unravel. Great. Metaphase I, the important thing to look at here is that we are going to have the pairing of homologous chromosomes, which again, that's where that bivalent terms comes in. That's where the synapsis happens. Great. So these pairs are going to move to the center to align like we are used to, except instead of just one of these, it's going to be 2, and they're going to pair together because they're best friends, aka they're both chromosome number 2. They might have slightly different DNA, and that's normal. That's actually what you want. And they are going to move to the center of the cell like so. Here is where crossing over will occur, right here in metaphase I, in meiosis I. It can only occur here, and I will talk about crossing over later. I keep saying that, I'm sorry. Well, I will talk about crossing over, but this is where it occurs. Moving along. Anaphase 1 is where we have one chromosome from each pair that is pulled to the opposite poles of the cell. So these friends that are like next to each other, kind of like this, one is going this way, one is going this way, and they separate. They're each going to be pulled to a different side. So now there are going to be 2 cells with 46 chromosomes each. Okay, so that's like the normal number of chromosomes. So right now we are still in the normal number of chromosomes. We're chilling. Great. So at this point, the sister chromatids, which again are going to be one half and the other half, they're still connected by the centromere, which is the little dot basically that we draw in the circle in the center. They're still connected at that point. Once that happens, telophase 1 is going to happen again. That's DNA is going to stop being supercoiled and it's going to unravel. The cell will pinch in, and now we have 2 diploid cells. Yay! Or 2, sorry. Oh, God. No, yeah, 2 diploid cells. And we're chilling. So let's go back to our statements. Now that I've kind of talked about that, let's see what we can check off. Again, I'm moving right past drawings. I don't feel like doing them, so I'm not. I think you can understand like what they look like what's happening. I did draw something. And then our next statement is DNA is replicated before meiosis, that all the chromosomes consist of 2 sister. Right. So I did mention this, but DNA is replicated in interphase before meiosis. So before meiosis even starts, that's why it's not under the prophase 1 subheading, the DNA is going to duplicate. This is going to mean that given a diploid number, for example, 2n equals 4, we can outline the movement and structure of DNA through the stages of meiosis. So if we're given a number and say we start with 2 and then it duplicates to 4, by the end of this we're going to have 2. And then by the end of meiosis 2, we'll have 4 sets of 1. It's confusing. That was not a good way to explain that. It'll make sense. It'll be fine. So let's move on. So again, cells are going to replicate in interphase before meiosis. So I guess I should discuss at this point what is a sister chromatid versus a non-sister chromatid. So I talked about how the sister chromatids are going to be connected by that centromere. Well, when we have chromosome 2 and chromosome 2 up here, the non-sister chromatids are going to be like this part and this part. This part. Or this part, right? Because it's on a different chromosome, even though it's the same genetic material, it's carrying the same genetic material, it is not a sister chromatid because they're not connected by that centromere in the middle. Okay. So, again, we're going to have our homologous chromosomes pairing up. 3 events that occur in prophase 1. Oh, God. Well, that nuclear membrane is going to like disappear. We're going to have the DNA supercoiling. I don't know why I wrote unravels. I'm losing my mind. It's going to supercoil, and then this is going to be the point when, I don't know, things start happening. Anyways, I'm doing great. So we'll talk about crossing over later. Again, we're going to have the bivalent and synapsis happening when the homologous chromosomes pair up. I put that in metaphase 1, but this is making it seem like that happens in prophase, so I'm not entirely sure which it is. I would say my metaphase 1, but yeah. Anyways, I will talk about crossing over later. This is all, sorry, I'm reading through our list of objectives, and they're all about crossing over, which I've decided to talk about later when it makes sense with linked genes. I digress. You didn't really need to know that. Sorry. Oh, okay. So we can talk about this. It is called independent assortment. So at this point in metaphase and anaphase, specifically in anaphase, independent assortment is going to occur. So basically, if you have two big chromosomes and two little chromosomes, and this is this is some homologous chromosomes. These are homologous chromosomes, and so are these. They have equal chance of this one going this way and this one going this way as they do of going in the direction, right? So this is called independent assortment because you can have any combination of your chromosomes going in a certain direction. So it's basically creating a random assortment, get it? Independent assortment of these 23 pairs. And this is going to create unique gametes. So if you're asked about in what ways can genetic variants occur, independent assortment is one, and this is what it is. Great. Independent assortment is due to the random orientation of pairs of homologous chromosomes in meiosis I. Random orientation is just going to be that how they're pulled in different directions, right? That's called the random orientation. Sorry, turning the page here. So this is meiosis I is both when the homologous chromosomes pair up and when they separate. So this is different from meiosis II because the sister chromatids in meiosis II, which we'll talk about in a minute, are going to be pulled in separate directions as well. However, they're not homologous chromosomes because they're not homologous chromosomes. All right, meiosis II, let's talk about meiosis II. So at this point, we have created two sets of 46. Oh, I'm losing it. 46, right? That's what we've created so far. In meiosis II, we're going to take these diploid cells because they contain two sets of 23, and we're going to make them into haploid cells. So we're going to end up with 4 haploid cells. The reason why we go from diploid to haploid at this point is because the DNA does not duplicate. So this is important to know that DNA is only going to duplicate in interphase before meiosis I and not before meiosis II. This is how we end up with a half set of DNA in each gamete. So in prophase II, that DNA is going to be supercoiling. I'm doing great. DNA is supercoiling, the DNA membrane is going to be disappearing, everything is fine and dandy. In metaphase II, the chromatids, which are still, by the way, connected with their little centromere, are going to line up in the center of the cell. The spindle is going to be attached to each of the sister chromatids, and they are going to be pulled to separate sides of the cell. So at this point, they say goodbye to their sister, and they both peace out. And then in telophase, no, that's right. In telophase, the DNA will uncoil, the cells will pinch off. All sperms, this is also important to know, is different, but all sperm cells are going to be the same size, but ovum cells are going to be the largest of the 4 for the female gamete. The ovum is going to be the biggest cell, and then the other 3 are going to be called polar bodies. So you can only use the ovum, you can't use the polar bodies. And the reason why it's this way is just to allocate resources, and the egg cell, here's a fun fact, is the only cell which we can see with the human eye, the naked eyes. You can see this ovum cell, but I'm assuming you wouldn't be able to see the polar bodies, they're too small. And that's why they're different sizes. It's just how it works out for females. But sperm cells are always going to be the same size. To compare meiosis I and meiosis II, there are no pairing up of homologous chromosomes in meiosis II. There's no pairing up of anything. Crossing over will not occur at this point. Sister chromatids are going to be pulled apart in Meiosis II, but that won't happen in Meiosis I. They're going to stay together because it'll be the homologous chromosomes that are pulled apart. Yeah, great. Oh, it's important to note that Meiosis I is what's going to be called a reductive division. This is because we are reducing the number of chromosomes from the amount that we had before. So yes, so the next part is that, the next important thing, I guess, to think about is that when we go from 46 to 23, again, we're going to end up with 4 cells. They're each going to have 23 chromosomes because we had duplicated before, and now we didn't duplicate, so they're each going to have 23, which is 1/2 of the regular amount. And then, I don't remember talking about this, and I don't have any notes on it, but there's also such a thing as allele segregation that occurs during meiosis, and the outcome says that the alleles of each gene separate into different haploid daughter nuclei during meiosis. So this is just kind of stating, similar, I'm guessing, to the ovum cell situation and the polar body thing. So the unlinked genes segregate independently as a result of meiosis. So unlinked genes are going to be the ones that are on different chromosomes. And again, they can be pulled in potentially different directions. I'm so sorry for all the text. I swear to God, I'm not popular. Sorry. Oh my God, I'm so out of it today. The unlinked genes segregate independently as a result of meiosis. This is, again, another way that genetic variation can happen. The sisters could be pulled in whatever direction they so please. So, crossing over we'll talk about later. So another thing that promotes genetic variation, if you're asked about that, is the fusion of gametes from different parents. So what do I mean by this? I mean, any of these random eggs that we've made or random sperm that we've made can fuse with any other sperm or egg. So that's totally random as well. It's the, again, that is going to be the fusion of gametes from different parents. So this also promotes genetic variation because you're going to have a variety of genes that are being put into the offspring. This is an important part of fertilization in the source of genetic variation. So the next thing that we're going to talk about is, well, let's see. I don't remember. Mitosis versus meiosis. So this will be quick. So we've talked about mitosis in the last video. If you haven't already seen that and you want a refresher on mitosis, which I've said this a few times, please go and watch that past video to get a better understanding of mitosis, but we will review it very briefly now. So mitosis, you're going to end up with identical daughter cells. The point is to make completely identical cells. But in meiosis, what you want is different daughter cells to promote genetic variation. Mitosis, you're only going to do division once, whereas meiosis, you have meiosis I and meiosis II, which means you're dividing twice. Mitosis, the result is going to be 2 diploid cells. So that's going to be 2 cells of 46 chromosomes. But in meiosis, you're going to end up with 4 haploid cells, which again, I didn't write that right. I did. Haploid is going to be 23. So you're going to end up with 4 cells with 23 chromosomes, which again, the chromosomes will be full for mitosis and halved by meiosis. Mitosis will make all somatic cells, whereas meiosis is only going to make those gamete cells, so either the pollen and the ova or the sperm and the eggs. Just a quick little overview for you there. So now let's talk about crossing over, which I know I've mentioned several times and we're all desperately curious on what it is. So meiosis is going to be a process that creates new combinations of alleles on chromosomes. So what do I mean by that? I mean that if we have a chromosome that's A and B, and another chromosome that's a and little b, which, by the way, if you ever see notation of a letter with a little line over it, I highly recommend that you write like that when we do Punnett squares or things like crossing over, because it just makes it clear that your letter is lowercase. Anyways, if we have this and they cross like they are here, because they are homologous chromosomes, this would happen during meiosis I, then we have the opportunity to get big A, little b, and big B, little a, and big B. So why is this important? Well, it continues to promote genetic variation. This is making it so that the chromosomes that the parent passes on are not exact copies of the ones that they have in their, that their chromosomes are. There's a chance that they will be totally different combinations within themselves. So the details that we should briefly mention is that the closer on the chromosome that the genes are, so like here and here, is very different than if it was here and here, right? So the closer they are, the more they are more likely to be inherited together. Because if you figure I go through and I cut right at the middle, the two at the top are going to be staying together, but the one on the bottom is going to be separate. This is because the two on the top are closer together. And then finally, crossing over deals with linked genes. So if you have unlinked genes, that means that they're on different chromosomes. So maybe chromosome 2 and chromosome 5. But linked genes is going to be chromosome or alleles on the same gene. So we can see that these are on the same sister chromatid here, or the same chromosome. That's going to mean that they're linked and that crossing over can occur. Again, this is going to be happening during meiosis I and will not happen during meiosis II. So we're going to come back and talk about non-disjunction some more once we go on to discuss genetic diseases or genetic issues. So I'll talk about that then. That is also on our paper. And then we'll go and do a pass through after, because I am checking things off that I talk about and I want to make sure I fit it all into this video. So let's move on to genetic inheritance, or what I've just called genetics. So the first statement is that Mendel discovered the principles of inheritance with experiments in which large numbers of pea plants were crossed. So let's story time, children. Let's all sit down and listen to a monk named Mendel. Mendel had pea plants, and he devoted his whole life to making more pea plants. And as boring and as excruciatingly dull a life that sounds, he was able to make some really, really important connections about genetics. All of the genes that he looked at were unlinked and obviously followed Mendelian law, which is the laws that he was able to decipher about genetics. And because of this, we were able to get a lot of information about unlinked genes, both monohybrid and dihybrid. And the first of that is dominant versus recessive. So let's go through a crap ton of vocabulary, all right? So, the first thing that isn't, or it is written there. So dominant is going to be alleles that will always be present in a phenotype. So these are going to be written with a capital letter like so. So if you have big A, little a, or big A, these are both going to be phenotypic dominant traits, right? Your phenotype versus your genotype is just going to be the traits that present. So if the phenotype is black fur versus white fur, and the black fur is dominant, then if you had either of these two genetic combinations of alleles, you would be able to see in the phenotype that you had black fur. Even though the genotype, the letters, whether or not you can explicitly see them in the phenotype, they will include one dominant trait, one dominant allele, and could either have another dominant allele or a recessive allele. So again, phenotypes are going to be traits that present in either a person or an animal or a plant or whatever. And genotypes are going to be traits or alleles of DNA. So the ones that you might not be able to see, but are still coded for in the DNA. This is going to be extremely important moving forward. So make sure that you have a good grasp on it. Again, recessive is just going to be represented by lowercase letters or little letters, and they're going to be needed. You need to have both little letters or both recessive alleles to be able to see that trait in the phenotype. It's important to keep in mind that while we consider recessive alleles to be less common, because you need both in order to present in the phenotype, there are some traits that are mostly recessive. I couldn't name them off the top of my head, but they do exist. So again, you need both little letters in order to see a recessive trait in your phenotype. So gametes, we've already discussed, but let's say it again. They're either going to be sperm, egg, pollen, or ova. They're going to contain half a set of DNA in each gamete. Oh my goodness, I have the serious hiccups. Zygotes are going to be the first stage of a fetus. So right after a sperm or egg cell, sperm and egg cells come together, they create what's called a zygote, and this is the first stage of life. Kind of, not really. But yes, that's going to be the first stage of a fetus. So next we have homozygous and heterozygous. So you might think that these words look very similar, and that's because they are. They are very connected. So homozygous, and you can think of this, we know homosexual means either a man dating a man or a woman dating a woman. And the reason why that is because homo means same. So we can remember that homozygous means the same type of allele because homo means same. So either this is going to be two dominant alleles or two recessive alleles. Whereas heterozygous, and again we can think of this by thinking about a straight couple or a heterosexual couple, a man and a woman, because hetero means different. Oh my gosh. This is going to be two different alleles, a dominant allele and a recessive allele. And this can also be called a carrier of the recessive trait. Because while you won't be able to explicitly see that recessive allele in the phenotype, it's still there. So if we're talking about a recessive trait and we're tracking a recessive trait, anybody that is that is heterozygous for that trait could be called a carrier. The next vocabulary word I'd like to discuss is autosomal. And this means that they are on a chromosome other than a sex chromosome. So not the X and Y chromosome. So the reason why this is important is because when we do look at sex-linked traits in the future, they are going to follow slightly different rules than autosomal traits. It's important to remember that unless you're looking at a pedigree, to basically assume, unless you have reason to believe otherwise, that we're talking about autosomal traits. So let's do a very basic Punnett square with a monohybrid cross. So let's look at the term called the generation, the F1 and F2 generation. So the F1 is basically going to be called the parents. They're going to be the first generation. I'm so sorry. In this case, we're looking at the two heterozygous individuals having children. So what we want to look at is what are the gametes that they could create? Well, let's look at one of them. So we know that they could either pass on a dominant allele or a recessive allele. So these are going to be the gametes that they could create. So when we do a Punnett square for a monohybrid cross, we're going to create usually 4 boxes unless you have a homozygous individual. Then instead of writing 4 boxes, you can just get away with doing, they're always going to have the dominant, they're always going to pass on only the dominant or only the recessive trait. And then if they're against a heterozygous individual, makes sense. So it can save you the trouble. If they can only create one type of gamete, then that is the easiest way to go about doing that. But in this case, we are not looking at that. We're looking at two heterozygous individuals. So we know that they can either create a dominant or recessive gamete. And so we put them in a Punnett square, and we can do the Punnett square and bring down the A, and bring over the A, and bring down the little a, and bring over the big A, so on and so forth. And we end up with these genotypes. So then what we can also look at is the genotypic and phenotypic ratios. So over here we have the genotypic ratios. We know there are genotypes because we're not talking about the trait itself, we're talking about the alleles. So you could have homozygous dominant, which we have one of. We could have heterozygous, which we have 2 of. Or you could have homozygous recessive, which we have 1 of. Therefore, our ratio would be 1 to 2 to 1. It is also important to say that you always want to simplify your ratios whenever possible. So if you have 2 to 2 to 2, you can simplify that 1 to 1 to 1. It's not always important, but it is kind of important to consider that possibility, right? So the other thing that we can look at is the phenotypic ratios. So we know that because these individuals, all 3 of them, have at least one dominant allele, the capital A, we know that they are all going to be presenting that dominant trait. Therefore, we are going to have 3 dominant trait individuals, and because this one is homozygous recessive, 1 recessive individual. So let's check off what we can about all right, I'm going to just assume that I covered everything here, and I'm just going to keep going. Keep on trucking along. I'm so sorry for these hiccups if you can hear them. But I'm not filming this again. All right, now let's talk about a test cross. This can be used for really any type of, well, not codominant probably, but it can be used for any monohybrid or dihybrid, whatever, right? So true breeding is going to refer to parents that are true breeding, and then they will always have homozygous recessive children. So what do I mean by this? I mean that true bred animals are always going to be homozygous recessive. We know that they're going to be homozygous recessive because they will present the recessive allele. It is not possible for an individual presenting with a dominant trait to be, we can't be sure that they're homozygous because there's a chance they could be a carrier for that recessive trait and have 1 dominant allele and 1 recessive allele. True-bred individuals are always homozygous because we are sure that they are, we're sure that they're homozygous because they're presenting that recessive trait, and so therefore they will always have homozygous children because homozygous recessive parents will always have homozygous recessive children, assuming both parents are homozygous recessive. So how can we use this information? What do we do when we know an individual is homozygous recessive or presenting with the recessive trait? Well, in this case, we can test whether an individual presenting with a dominant trait we can determine or attempt to determine whether they're heterozygous or homozygous dominant. So this is determining if an individual with a dominant phenotype is true-bred or not. To do this, we take a recessive individual, which we know is going to be true-bred because they must have 2 recessive alleles in order to present with the recessive trait, and we take a dominant phenotype individual, which could be either dominant or dominant recessive, and we breed them. This is helpful because we know that if the offspring are all a dominant phenotype, which means that they got the dominant, they had to have gotten that dominant allele from the parent that we're trying to determine. If all of the offspring come out that way, then we know that it has to be the case, or we know that it's likely the case that the individual we were test crossing for is homozygous dominant. However, if they come out with a phenotype child, with a phenotype that is recessive, then they have to be 100% have to be heterozygous because they had to have gotten one recessive allele from each parent. So we can never be certain unless we see a child with a recessive phenotype. In that case, we know that the parent that we were testing for had to be heterozygous in order to pass along that recessive allele. However, if we get all dominant trait individuals as offspring, and we do this many, many, many times, there's a very high chance that the individual is going to be homozygous dominant. However, we can never be certain because of the way probability works. There's always a chance that that's not the case and that they just had luck with probability over and over and over and over again. Let's talk about codominance. So an overview is codominance is when neither trait is dominant over the other, so both traits are present in the animal or plant or human. 2 examples of this is roan coats. Roan is going to be like when you see an animal, like a farm animal or whatever, that had white and red spots. Calico cats are another example. They have the tabby cat coloring, but also they're just multicolored. Being multicolored does not necessarily always mean codominance, but it is one example, right? So another important example to consider with this is blood types. Blood types specifically in humans, I assume. So, what letters do we use to represent the blood types? Because they have to be, or why are they codominant? Let's start there, I guess. They have to be codominant because type AB blood, right? Great. So, because of the way the markers work on the red blood cells, blood type is going to be codominant. So, the genotype for type O blood is going to be 2 lowercase i's. Type A can either be 2 capital I's with a subscript A, and that's what we call this, a subscript, with a subscript A, or 1 I with a subscript A and 1 lowercase I. The same is true for type B, except instead of subscript A, it's going to be subscript B. And then AB blood must always be capital I subscript A, capital I subscript B. These, I think, once you recognize that type O blood is going to be recessive, it's pretty simple. Like, you can basically just go off of what the letters say. If there is an A, then it's going to be type A. If there's a B, it's going to be type B. However, if there is type AB, then it's going to be type AB. And if you only have lowercase i's, then it has to be type O. Why they didn't just pick O, I don't know. But that's fine. So let's do a very quick example. So parent one has type O blood. So that means they have 2 lowercase i's as their genotype. Parent 2 has type AB. So that means they have a capital I with an uppercase, or with a subscript A and subscript B. So can they have a type A baby? So we're going to solve this the same way we would a regular monohybrid Punnett square question, right? So we're going to put the lowercase I. This is one example where you don't really need to do a full Punnett square. You can do kind of a half Punnett square. And then you can do the I subscript A, I subscript B, because those are the 2 gametes that can be passed on. So we will know that we must have one like this and one like this. So here is our type A blood baby, right? So the important thing just to remember here is that the only trait that's not going to be codominant here is going to be that type O blood. They're going to be lowercase i's. And that's really the main difference here. If you have any major questions on this part, which I can totally understand, I didn't really go in too deep with it, please feel free to shoot me a message or leave a comment and I will answer that. So let's talk about sex-linked traits. So remember previously when I said not autosomal? Well, this is why. So the definition of a sex-linked trait is going to be traits coded for by genes on chromosomes X or chromosome Y, but not both. So we're mostly going to see X chromosome, and I'll talk about that in just a minute. I already mentioned this. These are not autosomal. So X or Y is going to be your sex chromosome. If you have 2 X's, you're going to be a female. If you have an X and a Y, you're going to be a male. It is possible, because of non-disjunction, that you could end up with 3 X's or 2 X's and a Y, or I don't know, those are the only combinations. Or you could end up with 1 less. It is possible. It's not likely, but it is possible. Those individuals are called intersex. Usually, I believe. I think that this is specifically the type that's intersex. Anyways, more information you didn't need to know. So why do we see them more on the X chromosome? So these traits, these sex-linked traits, are more likely to occur on the X chromosome than the Y chromosome. And that's because there are more genes on the X chromosome. There are about 1,400 genes on the X chromosome, but only 200 genes on the Y chromosome. So that's interesting. But yes, that's why you're going to see it less often on the Y chromosome. It's also guaranteed that an X chromosome will be passed on to the child because they have to get an X chromosome from mom. And it is a 50-50, roughly 50-50 chance at any given point that the child will have an X chromosome or a Y chromosome. Because of this, it's going to be a lot more likely to see on the X chromosome. So the other thing to note with this is that because females are going to always have 2 X chromosomes, they can be carriers. This also means that in order to be affected, they have to get two affected chromosomes, 2 affected X chromosomes. So we, again, are using subscript here, lowercase meaning diseased or a trait that's sex-linked, and that's the recessive. And then again, capital is going to be the dominant. So affected females are going to be much less likely because you have to get an affected chromosome from both mom and dad. And then females are the only gender that can be carriers here because they are the only gender that has 2 X chromosomes. Whereas the males are going to have one X chromosome, so they are either healthy or affected if they have that recessive subscript. So in this way, because males can't have they will not have 2 of the traits, they cannot be carriers, right? So we'll talk about and examples of some sex-linked traits in just a little bit, but this is kind of the basics. So you can do your same Punnett square situation that we've been talking about this whole time, where you have a carrier female, for example, and let's say an affected male, and you want to know the chance of having an affected female. We can see right here what our phenotypic and genotypic ratios are. This is going to be our affected female. This is going to be our carrier female. This is going to be our healthy male, and this is going to be our affected male. So the only thing really to remember here, the biggest takeaway I'd say, is that you write these differently. The notation is going to be that X with the subscript A or other letter, and that these are going to be more common we're going to see them more common in males, but they are more common to occur on the X chromosome. Again, men can't be carriers. Always assume, for IB, always assume that when we're talking about a sex-linked trait, it is linked to the X chromosome. So let's talk about unlinked dihybrid crosses. I know, just what you wanted to hear about. So let's get started. So the definition is going to be 2 different phenotypes on different chromosomes. So they're not linked. So again, remember that linked alleles or linked crosses are going to happen on the same chromosome. But because these are unlinked, they're going to happen on different chromosomes. So I always go back to chromosome 2 and chromosome 5, just because I think they're fun. I could tell you one thing that chromosome 2 does, but anyways, moving right along. Mendel also discovered these. Back to our story time, when he did the pea plants, he looked at both if they were yellow or green, as well as if they were wrinkly or smooth, and so he was able to see unlinked dihybrid crosses. So when I mentioned the Mendelian law or the Mendelian ratio for dihybrid crosses, it is 9, 3, 3, 1. This is our ratio for if we are crossing 2 heterozygous individuals. So why is it important that we know this ratio? Well, it's just important, okay? It's just important. This is going to be the ratio that you're going to see for the phenotypes, not the genotypes. No, just the phenotypes. So here, I already did it because it's a lot of filling in, but here we have 2 heterozygous individuals for both traits. I wrote down their possible gametes, which again is going to be done using FOIL. So we have capital A uppercase B, capital A lowercase b, lowercase a capital B, and lowercase a lowercase b. So I wrote them again over here. And then I took these gametes and put them on my chart. So this is going to need a lot more boxes than our other chart. It's going to need 4 on the top and 4 on the bottom. Remember, when you line these up, you want to do capital, 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 and then lowercase, lowercase. But over here, you want to switch it. So instead of doing the first trait, you want to do the second trait is capital, capital, lowercase, lowercase. This just makes it so that you get an even spread. It's easier to understand the data. So when I say we have this phenotypic ratio here, where did I get it from? So this is going to be dominant, 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 right? Because they have at least one uppercase A and uppercase B. Dominant, 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 dominant. Sorry, not that one. This one. Dominant, dominant. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's how we get our 9. So then if we want to look at dominant recessive, we have 1, 2, 3. 1, 2, 3 of the recessive dominant. And then only 1 of the recessive. So what is the takeaway here with unlinked dihybrid crosses? Well, if you want to write out the Punnett square on your exam, do it this way. Do it with a lot of boxes. It's complicated, it sucks. Sometimes that's just how it goes. But if you're not looking to write this out for the exam and maybe you're answering a multiple choice question instead, another way you could go about this is by doing 2 separate Punnett squares. So we could do one for the A trait and for the B trait. And again, you would do this using the gametes. So we have capital A or lowercase a and then capital B or lowercase b. You could do them all out, A, B, 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 B. Sorry. And then you could like cross-reference them. Well, we know that we'd have dominant, 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 dominant. But I'd say it's probably just easier to go about it this way, unfortunately. Unless you're looking to find like whether a certain genotype is possible, it's easier to go about it with the big Punnett square, unfortunately. So then let's look at dihybrid linked genes. So linked genes are going to be traits that are on the same chromosomes. These would be both on chromosome 2 or on chromosome 5 or on chromosome 7 or any chromosome ever. They're just on the same chromosome. They're not going to follow Mendelian law. Mendel did not discover these. They don't follow his ratios, his 9-3-3-1 ratio, which just memorize that, please. They're not going to follow that 9-3-3-1 ratio anymore. But we can still look at them when we do chi-squared, which we are going to do probably next. So let's go over a little bit of vocabulary very quickly. What is a parental gamete or a parental whatever? So that's going to be the same as a parent. That means crossing over didn't occur. So I guess it's also important to mention, this is when crossing over would occur because we have the traits on the same chromosome. The further on the chromosome, the more likely to they're more likely to, whatever. Oh my God, I'm losing my mind. I know what I mean, you know what I mean. Closer together to staying together. Recombinants. So if we're looking for just the recombinant examples, recombinants are going to be the ones that are different from the parents. So you have parental and recombinant. If you're asked to find the recombinants, find any combination that isn't like the parents. And then you will probably not need to do this for the exam, but the recombination frequency, RF, is the recombinants divided by the total offspring times 100. So that's going to get you a percentage or a recombination frequency. That's That's basically just looking at how many of the individuals are recombined versus how many are not. And that's also going to be related to where they are on the chromosome, right? So if they stay together, they're more likely, if they're closer together, then they're more likely to stay together. So let's talk about the notation. So if you have those 2 bar lines, which we didn't see previously, but now here they are, those bar lines are going to indicate linked traits. So that means these 2 are linked and these 2 are linked. So the top is linked and the bottom is linked. So let's depict crossing over. So if we have gamete 1 and gamete 2, we have the little multiplication sign which shows that those 2 are breeding. You could either have, this is a parental, this is parental. These 2 are crossed over on the top because we can see it's different because we have capital lowercase or lowercase capital. So Why is this important when looking at linked genes? So linked genes are going to be written in this way. They're on the same chromosome. I already mentioned all that. It has to do with whether or not they're going to recombine. So recombination happens because of crossing over, and that promotes genetic variation. How many times will I say this in this video? 100,000, but it's important. So let's do chi-squared, everyone's favorite. Let me get my energy back for a minute. All right, so what is the use of doing chi-squared when looking at genetics? So we are going to be using it with dihybrid crosses to determine if the genes that we are looking at are linked or unlinked. So this is going to tell us if two genes are going to be on the same chromosome or not, linked or unlinked. So this is going to be comparing the data that we collect to that of Mendelian's ratio. Again, 9331, 9331, 9331, right? So to do this, we're going to use the expected value. So I have written out the steps here. I'm going to go through them, and I'm not going to read the steps aloud. Use your eyes to read the steps, okay? Okay, so at the bottom here, I have an example written out. I have the Mendelian ratio that I would be expecting for each of my data sets. I have the observed data values, which again is what we collected when we were doing our research that we definitely did. I have the total number of observed phenotypes. And then I'm going to fill in the expected by multiplying the Mendelian ratio by the observed value. So if I do this, I get 240. Pause now to try the rest yourself. Welcome back. So these are going to be our expected values. If this exactly followed Mendelian ratio, then these are the values we'd expect. With these, then we use the observed minus expected squared divided by expected to get, pause now to fill them in yourself. By adding all of these up, we are able to get our x squared value of 219. So this is a huge x squared value. So to find our degree of freedom, we're going to take our 4 expected phenotypes, subtract 1 to get 3. So our degree of freedom is going to be 3. We're going to be looking at the 0.05 column, and we see that the critical value is 7.81. It is always, for this situation where we're looking at dihybrid crosses, we have 4 phenotypes, is always going to be 7.81. It is important on your exam that you either show how you got there by writing out how you found the degree of freedom, and then what column you looked at, and then what the critical value is, or circle it on the chart that they'll give you. Go to 3, go to 0.05, and circle 7.81. It's always going to be 7.81 for this situation because we're using dihybrid crosses, and that's 3 and 0.05. It's always going to be 7.81. But it's probably easier for your life if you circle it. You will get points that way. So, because 219 is greater than 7.81, we know that the traits are linked, or that they are likely linked. Because again, this is science, and how can you ever be certain of something? So if you need any of the steps re-explained, please send me a message or write me a comment, and I will do that. So I've been mentioning this entire time, genetic variation, genetic variation, genetic variation. So who cares? Let's talk about discrete versus continuous variation. Discrete is going to have clearly defined categories caused by a single locus or location on the chromosome. So this might be This is going to be our monohybrid. Some examples are blood group, whether or not you have a widow's peak, and if your earlobes are connected to your head or not. Whereas continuous is going to be, just like in math class, a range of phenotypes. So this is going to be caused by a combination of many genes. This is called polygenic. And it can also be influenced by environmental factors. Some examples are height, skin color, intelligence, weight. So obviously, how do we see environmental factors playing a role? So for example, weight. Your weight is going to be partially determined by your chromosomes. It's a scale, obviously. You can find someone that's 90 pounds and you could go on, or you could find someone that's 200 pounds, very healthy. It can still be very healthy. However, if all you eat is potato chips and candy and junk food, grease, then that is one example of an environmental factor that's going to increase your weight beyond just what your genetics intended. So this is usually going to look like a bell curve shape. If you graph it and you do a histogram graph, you're going to find that it's going to look like a bell curve. So now let's cycle back and talk about genetic diseases. So these are going to be things caused by these are going to be unusual either mutations or whatever. They're going to be caused by genetics, so it's not like a disease that's you catch or something. They're going to be caused by genetics, might be due to non-disjunction in the case of something like Down syndrome, which I don't actually have on this list, but non-disjunction, again, is going to be when those homologous chromosomes are not fully pulled apart quite right. Then you will end up with non-disjunction and could have an extra chromosome somewhere. Yeah. So I have them listed out here, but let's talk about them just briefly. Sickle cell is going to be codominant. So you can have partially sickle cell blood or fully sickle cell blood. It's not sex-linked. Cystic fibrosis is going to be recessive and not sex-linked. Hemophilia is recessive and it is sex-linked. Huntington's disease is going to be dominant, which again, this is one example of where dominant is not the common trait. The recessive is the common trait because most people don't have Huntington's disease, but it's also not sex-linked. PKU is recessive, not sex-linked. And color blindness is recessive and it is sex-linked. It is much more common for males to be red-green colorblind. Yeah, so let's move on to pedigrees. Well, let's go back. Sorry. And I'm going to go through our statements very quickly, and we'll read through them together to make sure we've hit all of them. So, we've talked about dominant alleles. We've talked about genetic diseases. We've talked about how carriers for genetic diseases work. It's the same as regular carriers. We've talked about how some genetic diseases are recessive or codominant or sex-linked or not. We've talked about cystic fibrosis and Huntington's disease. We've talked about blood groups. We've talked about sex-linked traits. We've talked about red-green color blindness. And we're going to do pedigrees now. So let's just double check that we've done everything else while we're at it. Punnett square, yep. Predicting genotypes and phenotypes, yep. Chi-squared, yep. Predict using real data, yep. Chi-squared again, yeah. Non-Mendelian ratios we've discussed. Locus. So locus is going to be gene locus. It just means gene location. Location, you get it. We've talked about polygenics. We've talked about recombinants. We've talked about polygenic traits. And then going back to the front, we've talked about diploid nuclei. We've talked about crossing over. we've talked about schismata, we've talked about genetic independent assortment, sorry. We've talked about unlinked traits and how they segregate independently. We've talked about crossing over. And we've talked about non-disjunction. So the last thing we really have to talk about is pedigrees. Let's do that, and then we'll be done. And it'll only have taken like over an hour. So pedigrees, let's just go over them very briefly at first. So I'm not going to go real in depth with pedigrees. And the reason why is because I think they're kind of stupid. I think they make sense once you understand the basics, and as long as you remember the basics, you're going to be just fine. Circles are always going to represent females, and squares are always going to represent males. Why is this important? It's just important when we look at sex-linked traits and whether or not parents of whatever, right? Okay. So in the same vein, non-colored squares or circles are going to be the trait not being tracked, and colored in squares or circles are going to be the trait being tracked. Sometimes you will see a half-shaded square or circle or a dot in the middle of a square or circle, and that is going to represent a carrier. So again, that is an individual that is presenting with a dominant phenotype, but has one recessive allele and one dominant allele. So autosomal recessive versus dominant. So this is what I'm talking about here when I say that is I'm talking about how do we recognize if we look at a pedigree and there's not really any information, how do we recognize if that pedigree is dominant versus if that pedigree is recessive? If that pedigree is dominant, which means that the shaded or colored in squares and circles are going to represent a dominant trait, this is going to happen when affected parents or shaded parents can have an unaffected child. This happens if both parents are heterozygous or carriers of a recessive trait, then they have the ability to have an unaffected child. If unaffected parents or recessive phenotype parents are always going to have unaffected or recessive phenotype children, and an affected child must always have an affected parent. This is because the parent must pass on at least one dominant allele, and they have to present that dominant allele themselves. So if you think about the genetics and you think about how it works, this really should make a whole lot of sense. Recessive is going to be very similar. Again, this is when the shaded squares and circles are going to represent recessive traits. Two unaffected parents can have an affected child. So again, unaffected being not shaded, meaning dominant phenotype. They can have an affected child if those parents are heterozygous. If both parents are affected, which means both parents have at least one dominant, both parents are homozygous recessive, then the child must also be homozygous recessive because those parents only have recessive gametes to pass on. So I didn't really go into depth, I don't know why I didn't go into depth, on the sex-linked dominant versus recessive, but it's just going to be whether or not females and males are affected. We know that sex-linked traits are going to affect males more often. So if we're tracking a dominant trait, then it's going to be more females affected because they're going to have the dominant or unaffected or carrier trait more often. Then more females will be shaded, whereas with recessive, more males would be shaded because they're more likely to have the affected phenotype. All right, well that would do it for me today. If you have any questions, again, let me know. Leave a comment, send me a message. Yep, sorry this was so all over the place today. I really didn't mean for it to be like that, but it's just all over the place, okay? So I completely understand if you have questions. I know I didn't cover everything in intense depth, but we got a little taste for everything, I think. Yeah, that's it. Okay.